Thanks for the kind presentation. Good morning, everybody. I will start with a quite common first slide in the MPS, which is the need for a available, reliable, and predictive in vitro models for recapitulating the human biology in vitro. This is important not only for the pharmacological point of view, but also for the basic research, because we do need to have available predictive models for better understanding human diseases. And we typically aim to solve this issue by combining different skills, which are the development of organ-on-chip technologies, the validation, the internal scientific validations, and also the computational fluid dynamic modeling of what happened in terms of fluid flow induced and shear stress induced by the fluid flow within the organ-on-chip. And we have recently developed and validated this organ-on-chip named MIVO, Multi-In Vitro Organ Device, that has also received last summer the awards from the European Commission as Best Health Technology. And the main features of this organ-on-chip is that it allows to host and culture different types of cellular models, starting from the the simplest one, which is the membrane modeling, which is anyway very useful for many permeation purposes. We may, of course, host monolayer cells cultured in the current available inserts, also commercially available. This is important because many scientists appreciate to keep their well-established cellular models, just moving them in a physiological fluid dynamic environment. But of course, our mission is also to combine a fluid dynamic environment with three-dimensional cellular culture, meaning that we can culture spheroids, organoids, patient-derived biopsies, CHOPs, and many other, and also scaffold-based culture. Why we really believe that about the importance of the fluid flow? Because we have many publications, but also available coming from other groups worldwide, that prove the importance of the fluid flow in terms of their effect in many cellular phenomena, such as proliferation, differentiation, cytoskeleton orientation, but also focusing on the oncology and immuno-oncology, the importance of the fluid flow in terms of the capacity to affect the viability of cells in circulation, meaning both immunocells, but also most importantly tumor cells. So when tumor cells escape from the primary tumor and go in circulation, we have demonstrated that they may change their variability over time according to the different flow rates and shear stress induced by this flow rate. And why we believe it's important to keep the 3D tumor culture, because cells need to be cultured in vitro like in vivo. And this is especially true in oncology, because we have to try at least to recapitulate the intratumor heterogeneity. So it's really important from our perspective to maintain as much as possible the real complexity of the tumor environment. To do this, we typically work with a matrix. Typically, we work with alginate-based hydrogel, but it's not the unique choice. We have also worked with fibrin, matrigel, a combination of this, but this is just a proof of concept about the possibility to keep tumor cells in a 3D environment within an extracellular artificial matrix. But of course, there is a huge space to increase and improve also the complexity of the 3D environment, co-culturing different types of cells for really recapitulating the tumor model. We have worked with many different tumor models, starting from breast cancer, using different level of aggressive breast cancer cells. We then moved on a neuroblastoma pediatric model, again using different cell lines, and also ovarian cells. The alginate hydrogel has the positive feature that you can properly modulate the stiffness by adjusting the concentration of the alginate and the molarity of the calcium, which is the chemical cross-linker. And this is important because according to the cells that you use, you can change the stiffness of the matrix so that the cells may also display the proper spatial organization and cytoskeleton organization as we desire to obtain in vitro. Moreover, we also demonstrated that according to different stiffness, breast cancer cells are able to not only stay available and alive for longer time, but also to proliferate, migrate, create clusters like in vivo. Then we decided to combine these two important features, so the 3D and the fluid flow, by moving on a 3D culture of tumor cells in a MIVO organ-on-chip environment. We initially did an important comparison with the gold standard, which is still the xenograft model. So we used the same tumor, the same cancer, cisplatin cancer, the same ovarian cell line, and then we monitored the regression of the tumor in vitro and in vivo. And from a scientific point of view, it was also important to keep as internal control the 3D tumor culture in study condition. What we obtained, it was an important difference of the distribution of the proliferating green cells and on the dead cells in red. So what you see that, of course, not treated samples displayed a great amount of proliferating cells and very few dead cells, while in the dynamic condition, very few proliferating cells were observed after the treatment and a well and homogeneous distribution within the gel. These gels has a size of a couple of millimeters. While in static condition, you can see many proliferating cells also after the treatment, while the dead cells were only distributed around. So we concluded that we first hypothesized that the problem was the diffusion of the drug because we are working with 3D clinically relevant sites tissues, and then we first make a computational simulation of the diffusion of the cisplatin within the alginate matrix. Then we analyzed from a chemical point of view which is the real amount of the cisplatin within the matrix, and our hypothesis was successfully validated. So the issue was that in study condition, cisplatin was not able to properly diffuse within the matrix, so it is just able to kill surrounded cells that then create a barrier for the continuous diffusion. This is the reason of why we obtained the same regression of the tumor in mice model and in our organ-on-chip device, while the study condition displays a chemoresistance. And most importantly, the regression of the tumor was really faster, much faster in organ-on-chip, 75% of timing reduction. This is really important for pharma purposes. Then, happy to these results, we moved on. We changed the tumor model. So, since now I will speak about a neuroblastoma pediatric tumor model, we again created Howard gels in alginate. We evaluated the viability and the proliferation of two different cell lines. Then we checked also the capacity of these cells to respond properly to the typical drug used in this condition in clinics, and it was a proper dose-dependent response. Of course, in 2D, the conditions was different because the exposure of the drug of cells cultured in monolayer is overexpressed, as well described in literature. Then, immunologists involved in this work asked to check the proper expression of the receptors of the tumor cells, which are the target of the immunotherapies. So it was not sufficient to prove the viability and proliferation and the dose response of these cells. It was really important to evaluate the up and down regulation of the PVR, B7H3, PD-L1, and PD-L2 in both cell lines, and without and with the exogenous introduction of the interferon gamma that will be then released automatically by immunocells. And it was really, really important to observe the proper down regulation of the PVR and the upregulation of the other receptors, and most importantly, the upregulation of PD-L2 and B7H3 never observed before in these cell lines in using the standard conditions. So we then decided to culture this tumor model with the proper immunophenotype within the organ-on-chip chamber. So the tumor model was placed within an insert whose membrane has a pore size of 8 micrometer in order to avoid any physical limitation for immunocells infiltration. And the immunocells were placed in circulation. And in particular, we used a flow rate which is in line with the capillary blood flow condition. So we had immunocell circulation here, mimicking what happened in vitro, a barrier, a physical barrier, and then on top the tumor niche in an extracellular matrix. It's important through this picture to highlight the fact that it's possible to resemble different and separated niches in the same organ on chip. So in case it's important to use different growth factors, different media, or in other applications, different pH, it is possible to maintain this gradient in the donor and in the receive chambers for days. We analyzed the viability of the immunocells also in 3D condition, because it was never done before using this fluid dynamic condition, and the viability when the cells are placed in a 3D condition is fine. So this was important for what I will show later. Moreover, these natural killer immunocells we have used display the proper CD16 expression. This is also important because we will use this marker to decouple the signal of the tumor and of the immunocells. Then the first important result was the measurement and the quantification of the immunocells able to infiltrate the tumor niche, meaning the tumor cells that are able to win the gravity force and win the drag force, which is the viscous force created by the fluid flow, and enter into the tumor niche. So we also analyzed and carry out computational fluid dynamic simulations that prove that there is really a protrusion force, a biochemical force, that allows to some immunocells to escape and infiltrate the tumor. And most importantly, we had as negative control, of course, the absence of the tumor niche, so just the media. But we also used an important positive control, which was the same matrix without the tumor cells, with an increasing of 30% of serum, so an important chemoattractant, although also in this case we observed a significantly higher infiltration of immunocells that have left the flow and were counted in this niche. And most importantly, these percentages are also in line with the data obtained in clinical. We then analyzed within the gel what happened. So in the gel we observed some immunocells starting to interact with tumor cells. So in green here immunocells, in red the tumor cells, and it was so very important to observe not only the infiltration in the upper side, but also the capacity of these immunocells to enter into the matrix, migrate into the matrix, and going closer to the tumor cells. And moreover, we also analyzed the apoptosis of the tumor cells if compared to the standard conditions, and the apoptosis of tumor cells was significantly higher. So not only infiltration in the upper side, not only infiltration in the tumor matrix, but also an initial activation of the immunocells and killing of the tumor cells. Moreover, I will not show the data here, but we also checked the phenotype of the immunocells, and what we obtained is that they were less CD16 positive, meaning that the immunocells that enter into the tumor were more the CD16 negative. This is exactly in line with the clinical data. This is not a nice result, because we know that the most promising immunocells to kill the tumor cells are the CD16 positive, but from the clinical we also know that unfortunately the cells that are able to enter into the tumor or the pediatric tumor are the 16 negative. The capacity of the organ-on-chip technology to generate also multi-organ connection open the possibility also to create the primary tumor condition in one niche and the secondary tumor target of the metastasis in another niche. So we can really complete the overall journey of the tumor that may migrate in this direction, and also of the immunocells that may hopefully kill tumor cells. And you can also use this platform, of course, for testing immunomodulating agents. You can see here also the possibility to functionalize and cellularize the insert, meaning that in other works we have also endothelialized the insert with UVEC cells, so you can also increase as much as possible the complexity and the predictability of the model, creating also a vascular barrier where the tumor cells grow in contact. The three-way valve of the MIVO device allows to sampling the media for different type of analysis, molecular, genomic and chemical analysis. And we are also working with some partners about the possibility to culturing directly the patient-derived biopsies, also for testing personalized therapies, and for a scientific point of view, for better understanding why in some cases immunotherapies approach aims to combine physiological fluid flow for more predictive assays using 3D tissues also towards personalized therapies. And last but not least, I thank the group of React for Life, the CNR at the University of Genoa and the Ospedale Gaslini of Genoa, and of course the European Commission for the grant. Thanks for the attention.